Good day, everyone. For this lecture, we will be discussing two virus families, Asfarviridae and Iridoviridae, starting with the former, right? Asfarviridae is known for its only member, the African swine fever, which farm veterinarians in the Philippines are currently battling continually already since 2019. And it's quite hard how to do that, and it is still a challenge for everyone. And we can see the repercussions of the pathology of African swine fever with the rising prices for pork. All right, let's start with the properties. African swine fever is the sole member of the genus Asfarviridae. This virus is an enveloped, quite large, 200 nanometer in diameter. It has a complex icosahedral capsid, as you can see right there. And within that capsid, there is a central hole, which contains the genome, which is a single linear double-stranded DNA. So if you are wondering why is not doc saying negative-stranded or positive-stranded, when you have a double-stranded nucleic acid, you automatically have one being a positive, one negative. All right. Now, ASV viruses are thermolabile and they are sensitive to lipid solvents, which is usually what we use as disinfectants. They are resistant at a wide range of pH from 4 to 13, and they can survive for months and even years in refrigerated meat. That is why, they can be destroyed by a lot of heat and what do you call this? Cooking and heating. And African swine fever is not zoonotic. So that is why a lot of people are not buying pork because they think they can get the virus. No, it's not that. They're not buying pork because pork is so expensive right now, because when a pig dies from ASF or any other disease, we call it botcha and double dead, you cannot sell it for human consumption anymore. The thing is, why it is spreading like wildfire right now is because you may not be able to sell it to humans for human consumption, but you can slaughter it and feed it to other animals, which actually, when you say kaning baboy, it usually contains those offal and meat from dead pigs, right? And of course, you would not expect that to be a normal thing to be done in commercial pig farms, but backyard pig farms are a different thing, right? So host range would just be pig species, so domestic pig, the wild boar, warthog, bush pig, right? And transmission would be direct contact with the fluids or secretions of a sick pig, ingestion of meat. They say that when this virus goes into those dead ends like brain, some muscles, that is like the end of transmission because they won't shed like secretions and excretions. But when those dead ends are ingested by susceptible hosts, then virus spread happens. And they are found to be transmitted by arthropod ticks, the Ornithodoros ticks. All right, so what is the cell tropism for ASF? They would go for the monocytes and macrophages, which are key players in the immune mechanisms of the animal. They replicate both in the nucleus and the cytoplasm. The viral DNA synthesis is done or is happens in the nucleus. They say that after infection in the laboratory setting, you could actually see traces of the viral DNA in the nucleus already after an hour. The virus assembly is happening in the cytoplasm. All right. So entries through RME, receptor mediated endocytosis, although some would also point out other forms of endocytosis like pinocytosis and phagocytosis. Two proteins have been found to be active in virus attachment, P72, P54, and two minor proteins, P12 and P30, while P30 is more focused on virus internalization, or how the virus is inserted into the cell membrane. The genome carries all the genes coding for all replication proteins, meaning it is using its own virion-associated enzymes to conduct replication, right? So what else? The virus can also form this, what we call, consider here, just the nucleocapsid housing the genome, right? So again, like pox viruses, they do not need an envelope to be infectious. They can exit through budding or through lysis. Sorry, I did not put it there. Again, they do not need the envelope to be infective because the cell, the cell receptors that they could hijack can also be associated or attached to the capsid proteins themselves. All right. Now, patterns of transmission, you will be focusing on this one, I think, in med, because this would be a very good example for medicine subjects. All right. There is two basic patterns of transmission. You have the domestic cycle and the Sylvatic cycle. In the Sylvatic cycle, the African swine fever virus is maintained in asymptomatic wild pigs and in those soft ticks, Ornithodoros, argacid ticks, which occur in burrows used by these animals, right? So the ticks are biological vectors of the virus. After it spreads on viremic swine, the virus replicates in the gut of the tick and subsequently infects its reproductive system, which leads to transoviral and venereal transmission of the virus. It also can be transmitted between developmental stages, of the tick. It's what we call transstadial transmission, and is excreted in the tick saliva, coxal fluid, and the Malpighian tubule excrement. Right? So the infected ticks may live for several years. They can survive for long periods of time without a blood meal, and they are capable of transmitting the virus to swine, right, at each blood meal. That is where we go to a domestic cycle, that once the virus has been transmitted into domestic swine, either by bite of infected ticks or ingestion of meat or offals from infected animals, then that is what you call a domestic cycle. Okay? They would shed the virus in high titers, high amounts in the nasopharyngeal excretions and during onset of the clinical signs, and viruses also in other excretions like feces and urine during acute disease. Transmission of ASF is through direct contact and within buildings by aerosol. That is why it is very much of a big problem right now for swine practitioners on how to protect their flock from ASF virus. They do strict biosecurity measures, but again, mechanical spread through fomites like people, vehicles, are of very high potential to cause spread of this disease just because of the stability of the virus in swine, blood, feces, and tissue. So imagine if you're not cleaning your boots, if you are not changing your clothes, you're not doing the showers before you visit another farm. So that's a big problem. And the thing is, if our own herds of pigs are decimated by this virus, we are going to rely on imports, which is not good either. All right. So ASF is also known for evading and modulating the immune mechanisms of a susceptible host. Its target cell would be the macrophages, and some of the genes within the genome of the virus have been found to cause certain immune consequences. The A238L gene encodes for a protein which inhibits the nuclear factor kappa beta, which downregulates the expression of cytokines, which are controlled by that cytokine. So that's tumor necrosis factor, type 1 interferon, and interleukin 8, right? And this gene, the A238L gene, can also act as an analog of cyclosporin A. And if you don't know, cyclosporin A is a very good immunosuppressive drug we usually use for a lot of diseases wherein the immune system is too fired up, right? It also plays a role in expression of fatal hemorrhagic disease in domestic pigs, but only causes mild disease in the natural host, which is the warthog. And another protein is the 8DR gene, right? Or what we call the PEP402R, which you can see there, right? This is basically acting as a viral homolog or kagaya ng cellular CD2 involved in T lymphocyte activation and hemadsorption of infected cells. Right? So pathogenesis and pathology. What makes this hard to diagnose is that it shares clinical signs with hog cholera. Right? It attacks the immune system, which basically renders your animal incapable of fighting the virus and makes it more susceptible to secondary bacterial infection. When it replicates in the respiratory system, it goes into pharyngeal tonsils and lymph nodes, draining the nasal mucosa, which leads to primary viremia, using the RBCs and WBCs as Trojan horses, causing generalized infections and shedding in all excretions and secretions. All right. Gross lesions, you would have hemorrhage, particulation of serous surfaces. That kidney is well known for the turkey egg appearance, but that is for hog cholera, right? For chronic diseases naman, if it's not acute or if the animal has been fighting it off for a while, those lesions would go into cutaneous ulcers. It will cause pneumonia, inflammation of the heart, inflammation of the lung tissue, and arthritis. All right. So this is a very economically significant disease that we hope farm veterinarians back home would be able to to control soon, because we cannot keep on relying on on imports, right? It's not. We have to be agriculturally sustainable. That's the term, right? So that is it for Asfarviridae, right? Let's now go to this picture. I forgot. This shows the Particulation and hemorrhages in pigs with African swine fever. You have subcutaneous hemorrhages of the ears starting at the tips, followed by generalized hemorrhages on the body, enlargement of the gastrohepatic lymph node, enlargement of the spleen, because if you have done necrosis, or study the anatomy of the pig, the spleen is very, very small, right? So let's move on to the other family, which is a sister family of Asfarviridae. This is Iridoviridae. As you can see, morphologically, they're very similar. You have that icosahedral capsid. The genome is protected by an inner membrane. Then that's where the capsomeres would also be covered in. And sizes are not that different too. Right? Again, same. Single, linear, double-stranded DNA. The genome, the thing is, the genome of vertebrate iridoviruses is highly methylated. The invertebrate viruses are only 25% methylated. And why is this relevant? All right. Di ko pa pala na-discuss. Iridoviruses are known for affecting reptiles, frogs, tadpoles, salamanders, newts, and such. All right. So not much into fishes, and not much into the mammals, such. So why is there this methylation? Methylation is just the process of putting a methyl group within a DNA sequence. So for vertebrate, they're highly methylated. Invertebrate iridovirus is only 25% methylated. This happens in the cytoplasm, and they say that the DNA being methylated protects the virus from endonucleases, meaning enzymes within the cell, which could attack the virus nucleic acid once it is inside the cell. Remember, upon entry, they are shed, the envelope are shed, it is uncoated, then it goes into where it is replicated. And while it is being transferred, a lot of the endonucleases within the cell meant to defend the cell can attack the virus nucleic acid during that transport. So to be able to evade those protective mechanisms of the cell, you have methylation. All right. So what are the members, the genera within the family Iridoviridae? You have basically 5: the Iridovirus, Chloriiridovirus, Ranavirus, which is the most common that I know, Megalocystivirus, and Lymphocystivirus. And you also have 2, which are unclassified within the 5 given genera: the white sturgeon Iridovirus and the erythrocytic necrosis virus. Alright? And dito mo makikita actually yung the way that we name viruses with how related to the pathology it causes. Right? So replication first. Replication. Very similar to ASF virus replication, comes in through receptor mediated endocytosis and membrane fusion eventually. Replication is nucleocytoplasmic. A part of it happens in the nucleus, a part of it happens outside. See, first stage, second stage. The methylation happens in the cytoplasm as well, and that acquisition of the envelope is not required for a virion to be infectious. All right, so those viruses which exit through budding are exiting as enveloped. Some would go into another way wherein they will not get an envelope but they are infectious already, and they can exit the cell only through cell lysis. All right. The exemption as compared to Asfarviridae is that they use the cell's enzyme. For Asfarviridae and Poxviridae, they carry the genes encoding for the enzymes. So those are virion-associated transcriptase. However, for Iridoviridae, they use the cell's enzyme, okay? And they modify it to favor the synthesis of viral mRNA, okay? So let's start. That is basically. So the Lymphocystivirus affects basically fishes, freshwater, and marine. They're benign, self-limiting. They would again just be causing cutaneous problems, right? They would cause hypertrophy of fibroblasts. You can see the fibroblasts are usually like spindle shaped. Now it's so big. They have what do you call this? They develop this distinct hyaline-like capsule. You have enlarged nucleus, and you have the cytoplasmic inclusions, which are irregular in appearance. So this is what you call lymphocysts, and this is grossly, they look like this: hypertrophy of the skin and the gills, right? So transmission is direct contact or direct contact with this from fish to fish, or the fish sheds this hypertrophied part of the skin and gills and then gets in contact with another fish within the same habitat. The consequence of this is that it doesn't really cause, the productivity level basically ng fishes. However, it is more of a loss of commercial value because of the cosmetic appearance, right? For food, because you do not want to buy something with that, and for ornamental fish as well. So prevention for this is early diagnosis, a separation of fish populations, and if you find an infected fish, you quarantine it, right? So megalocystiviruses, just by its name, megalocysti, big cells, cytomegalic, kumbaga. One notable virus from this genera is the infectious spleen and kidney necrosis virus. It causes these cytomegalic cells. You can see right here in internal organs. The macrophages as well would have cytoplasmic inclusions, and as we know with cytoplasmic inclusions, those are indicative of viral assembly sites. So what are the internal organs? Usually go for the spleen, the kidney, intestine, even the eye, pancreas, liver, basically everything, right? So next, Rana viruses. We're going to discuss briefly, honestly. Three viruses within this genera. You have the Epizootic Hematopoietic Necrosis virus, which you might think it's so minor because it only affects fish, but that actually appeared during my licensure exam, so you just don't know what to remember. All right. So by its name, it causes extensive necrosis of the liver, the pancreas, the cells of the kidney, the spleen of fishes in freshwater or saltwater. The Ambistoma tigrinum virus, which is actually the called the scientific name for a certain species of salamander, right? It usually affects both larval and adult stages, and they are exhibited as combination of necrosis and hemorrhage. And the thing is with this virus, it's quite sensitive to the temperature of the salamanders. For they are infected at around 26 degrees Celsius. They survive usually, but if those salamanders are in a lower temperature, which is 18 degrees Celsius, they mostly die. So they don't know the role of that yet. It's still under researches. They don't know if the virus actually works better with lower temperature than in higher temperature. And the last one, which is the first rhinovirus to be discovered, is the frog virus 3, right? And it's not just affecting frogs, it's affecting larval salamanders as well and newts. And it causes petechiae and necrosis of the skin and parenchymatous organs. It can also be seen from the outside, as you can see here. And it can be transmitted through direct contact and fecal-oral contamination. Now, this is why these families are discussed together, because they share the same pathology. Genomically, they also share some characteristics, and they're quite so small as compared to other virus families. All right, this is the last slide for this lecture. Yes, this is quite short, which I think you are thankful for. You're now leaning on your chair and you're saying yes, right? This is again, this one is already covered in the second lecture exam. We are not going to include in the first lecture exam. So yeah, chill out. I don't think we will be, because it's Holy Week next week. I don't think we will be having any class then. And then after that, it's 1 week, and then it's midterms. So we'll see. All right, we'll see. If you're getting lazy already, I'm getting lazy too. But we have to stay strong for that. All right. Thank you for listening. Send me any questions that you might have. If you are interested with ASF, there's a lot of articles and notes and videos online. Just make sure what you're watching and reading is, reputable source. All right. Thank you and have a nice day. Bye.